Bhattacharya. So, Dr. Bhattacharya, can you?
Yeah, I am here.
Yeah. Okay. So,
yeah.
fine. Yeah, thank you so much for, I hope I didn't keep you waiting, but it was like, the session was engaging.
Nice
Yes, absolutely. So I would like to welcome Dr. Shubhendru Bhattacharya. I also call him Shubhendru. So he's a very old friend of ours. So as I would like to let you know, Dr. Bhattacharya, that this is a faculty development program, and over here we have faculty, students, all of them participating in this kind of a program, wherein they can really be directly engaged in putting forward their doubts and their questions in this session. So here I take the pleasure and honor to welcome Dr. Shubhendru Bhattacharya. He is currently the senior principal scientist and the head of the department of the molecular biology and biochemistry unit of Indian Institute of Chemical Biology, CSIR, IICB, Kolkata. And also he's the ACSIR Swarna Jayanti Fellow. Now, Dr. Bhattacharya is engaged with the IICB for a pretty long time, and he is actually most famous to be known as an RNA biologist in our country. So this is a very, very proud moment for us, because we have really such great stalwarts amongst us. So Dr. Bhattacharya, he completed his undergrad and master's from the University of Calcutta, from the Department of Chemistry and Biochemistry. And then he completed his PhD from IICB. So he's a son of soil, our own Kolkata. So it's wonderful once again. Now, after completing his PhD in IICB, he moved to Basel, Switzerland. And he got his postdoctoral training from the Friedrich Miescher Institute, FMI, in Switzerland. And over there, he made exceptional and outstanding breakthroughs in research, and found out the mechanism, trying to understand the mechanism of the activity of microRNA modulation in the mammalian cancer and immune cells. So that was another aspect that we are going to listen today. He has been the recipient of several awards, a long list of awards that we can see. So he has the AASS Young Scientist Award, the INSA Young Scientist Award, the EMBO Long-Term Fellowship Award, and then the CDA Award, and the International Senior Research Fellowship, and the Wellcome Trust. So there's a long list of awards that Shubhendru has, but the most commendable one, which I'm really proud to announce, that in 2016 he was awarded the Shanti Swarup Bhattagar Award. That makes us even more proud, and then followed by this National Bioscience Award. That was also another one. He has received the Nasi Scopus Young Scientist Award, and as I've already mentioned, the Swarna Jayanti Fellow. He's already a Swarna Jayanti Fellow, and most recently he has been recognized by the Khosla National Award and Sun Pharma Award in medical sciences. So I do not know how much more he has going to. There's a lot more we can expect in down the line. But Shubhendru has made all of us very proud. And today he is going to talk on the systemic gene tagging by CRISPR-Cas9, a new tool in understanding cellular organization and function in higher eukaryotes. So I will request all the participants to again, please give your full attention, and it would definitely be an engaging session once again. And thank you once, Dr. Bhattacharya, for giving us this precious time of yours. And now it's over to you, and the stage is yours. Thank you.
Thank you, Jayadi, for the kind introductions. I'm fully audible, I hope. And
yes,
yeah,
absolutely.
So, as Jayadi mentioned, that my lab actually works on different kinds of RNA, not the CRISPR-Cas9 per se. It's microRNA because or small RNA, but to keep the motto with the session and other things with this particular topic that I'm going to talk about, I'm probably not going to touch anything on microRNA. I only tried to talk about some kind of CRISPR based things that you already heard some of it, but maybe I try to highlight some of the new aspects that that may not have been covered by the others in this session. So I'm just going to share my screen. Hope I will able to. Let me find it. Yeah, is my screen is visible?
Yes, sir.
Okay, now I'm going to the full screen mode. So let's see. So I think it's now full screen and
Yes, sir. Yes, sir. Yes, sir.
And I am changing slide. You can see the change slide.
Yes,
also.
sir.
Okay, so that's fine. So I'll try to try to talk about the systemic gene tagging, as Dr. Bandyopadhyay already mentioned, that how this CRISPR-Cas9 techniques can be used to to tag some genes in the system, and how this can be a useful tool to understand the cellular organizations and the function of in higher eukaryotes. So that's in the nutshell that I'm going to talk about. But before that, I'd like to give a brief introductions about the CRISPR-Cas9 based system and all other Cas also little bit, and then I'll like to touch upon the basic application of the CRISPR-Cas9, but maybe already covered, and some of the mechanistic aspects, how it works. And then I'm going to talk about some of the application orientations, particularly in the cell biology field or molecular biology field, how it open up. So any questions that would come in your mind, please put into the chat box and try to answer at the end of my talk. And on the way, if you have any other question related to any other RNA, like even microRNA, if you find some question important for you, I will try to answer as much as possible. So with that, I go into the next slide. So as you all know, the CRISPR stands for the clustered regulatory interspace salt palindromic repeat, and it's a family of DNA sequence that originally found in the genome of prokaryotes like bacteria and archaea, which uses it in the antiviral defense mechanism. So when the virus invades the bacterial cell, there is an adaptation phase. When the new RNA sequence or the DNA sequence from the bacteriophage or the viral virus infecting the bacterial cells that get inserted into the CRISPR locus, and this new spacer is derived from the virus and targeting the CRISPR sequence. That is called part of the adaptation process. And the production of CRISPR RNA happens when this locus get transcribed to the CRISPR RNA is formed, and then it forms a complex with another proteins factor, in this case Cas9 protein. And this CRISPR RNA guides the molecular machinery to target and destroy the viral genome, which has the complementarities to this CRISPR RNA. So future targeting of the bacterial cell by any virus will ensure that the virus cannot kill the bacterial cells, and in that way the bacteria should be immune to this virus infections. So this adaptation, production, and targeting, these are the three phases that the bacteria machinery uses to protect itself from the invading pathogen like bacteriophages. So when you talk about the Cas9 protein, is a CRISPR-associated endonuclease, which is an enzyme acting on the molecular scissors to cut the DNA at a location specified by the guide RNA. The guide RNA is an RNA element that comes from the CRISPR locus, which actually find the complementary to a part of a double-stranded target DNA, and a specific site-specific cleavage actually occurs just upstream of the complementary sequence, and in that process, the double-stranded breaks then try to get repaired, and in the process of repairing, some kind of insertion may happen, some kind of mutations may happen, and in that way, It's a versatile tool to in the gene editing technology, which is used to modify, delete, or correct the precise region of a DNA of any organism in that sense, if they are expressing the Cas9 and the guide RNA. So again, I just to recapitulate how CRISPR-Cas9 work. The Cas9 form a guide RNA complex. This complex attached to the mismatched genomic DNA sequence and adjacent to the spacer regions, in this case in yellow. And this Cas9 RNA complex cut the double-stranded DNA, and in that way, a programmed DNA inserted in the DNA inserted in the cut. This is the programmed DNA, so that can be put into the positions to create a mutated sequence or a sequence with an alteration. In that way, CRISPR is useful to introduce any foreign DNA into the system or remove any unwanted gene from the system. So that's why it's a useful way to modulate the gene targeting. So that's where it go. And if we go to the history of how these things evolved, what time, so one can see in 1987 the first report of CRISPR cluster repeat was there, which is the term coined the CRISPR in 2002. It takes a long time, a long number of publication was there in between that resulted this kind of interest in the field. And then in 2007 only the first experimental evidence of this CRISPR conferred adaptive immunity in bacteria has been there. And then the discovery of the CRISPR CRRNA, the functional CRNA was there because it's a part of this CRISPR machinery. And tracer RNA was discovered in 2011, whereas the CRISPR in vitro study was done in 2012 because for its efficiency and gene cutting method. So in 2013, there was a very interesting breakthrough when this technology has been used in the mammalian cell to find they can cut the mammalian genome as well. So discovery of the dCas9, which is a Cas9 defective mutant, CRISPR-I or CRISPR-A. Well, there I would going to touch upon these things in the later part of my talk. And there is a genome-wide functional screening with Cas9 has been performed in 2014 and so on. And Cas12 was discovered in 2015. The invention of base editors, Cas13A was there in 2016. Also, in 19, the invention of NCAT and CRISPR-9 Cas9 system was there. In 2020, discovery of the very first CRISPR was there. So these are the different development we are seeing over time. These are not only in the context of bacterial genomes and machineries, but it's also in the context of its application, which is much gets I would say acceleration in the late 2010s, and then so on. It's going on. So these are all interesting things we can think about. And if you look into the number of publications we're getting out of this field, is very interesting. 1987 was the first report was there, but when you compare it in the 2016 onwards, you can see there is a huge amount of publication is there in the PubMed, over 4,000 per year almost that are there. The report is from 2019, a paper published on that. So you can imagine that how people getting interested into the field and how many papers are getting published over time in that particular topic. So that's itself tell us about the importance of the research in this field and importance of the techniques as such in gene editing problem, and which is much more recognized in the Nobel Prize given in chemistry in 2020. You all know that Emmanuel Serpentine and Jennifer Doudna received the awards for their outstanding contribution for the development of a method of genome editing that is very fascinating, because as we all know that is a microbial immune mechanism which is there to tackle the viral infection of a bacteria are now being used to edit the human genome even, and they have promised to have solution for different diseases. That's really fascinated the whole community and science community, and that's why they have recognized by the Nobel Prize. So as I mentioned, this technology has now already got space in different kind of applications. They have a lot of advantage, like this technology is very faster, cheaper, simple, accurate, and reliable, whereas the wide array of applications is in place. For example, if you talk about elucidating and epigenetic edits that can be done with this technology, it has application in the agricultural and food technology field. It has application in the eukaryotic and for genome editing in the context of fetus, such that some genetic diseases can be tackled. So nucleic acid imaging, detection, and diagnostic, which I'm going to talk more on that particular aspect today, is there in with this CRISPR-Cas9 techniques. So you can generate animals with specific phenotype or specific trait that called cell and animal models that are there. And more importantly, the human disease therapy, they are really much more in use with this CRISPR technology. And not only that, this technology is useful to do some new drug screening with the help of this CRISPR-Cas technology. So you can imagine that whole spectrum of biological research are getting affected or getting improved by the help of this technology. So that's why it's very important to know. So when you talk about this evolution of these gene editing techniques, so there are meganucleases that are used by certain lower organisms. They have zinc finger nucleases or TALEN. They all are based on recognition of some sequence where these in nucleases can come. So apart from the nucleus moiety, they should have a recognition motifs of the protein that should be present on that particular protein that make them more specific. For example, the meganucleus is less specific than these zinc finger nucleases. They recognize the DNA with triple nucleotide sequence recognized by this kind of domain of the proteins, which is TALEN is also using the same, but it's recognized a longer sequence, for example. So a little bit more specific than the zinc finger nucleases, but the CRISPR-Cas9 is the most evolved one as far as specific gene sequence identification is concerned, because it uses a specific RNA sequence that is much longer than the TALEN and zinc finger nucleus recognition motif by the protein. So this RNA recognize the target sequence where this Cas9 nucleus should bind, and then it cuts the introduce a double stranded break, and then the non-homologous enjoining may happen, or template edited homology directed repair would happen to have a specific sequence of DNA to be inserted. So you can see the evolution of this kind of technology, which have two parts. One part is the recognition of the specific sequence, and then the internucleotide cleavage. So recognition can be done through a protein domain, like for the zinc finger or TALEN, but it can be done through an RNA itself, which is in the case of CRISPR-Cas9. So that big give them the more specificity than the other systems that have been evolved over time. So CRISPR is the most efficient and feasible among the tools that are available for the gene editing as such. So when you go into the guide RNA prediction concept, there are two kinds of CRISPR system could be present in the bacteria. There are actually have more than two. So in the type 1, specifically, they have a long list of Cas9 protein that are expressed from the locus, along with this pre-CRRNA, and this CRRNA is processed in the Cas6, the CRRNA of desired sequence, with the complementarity to the specific DNA sequence, and they, with the help of Cas9 and other Cas, they actually recognize and target the cleavage of the target genes. On the other hand, the type 2 uses only the Cas9, the bigger one, which encoded also a tracer RNA, and this tracer RNA and the CRRNA both form a complex CRNA tracer RNA complex, and that the Cas9 uses to target a specific sequence. So bacteria also have a different mechanisms and different Cas locus to target differently to the genome of a virus or so. So that's why they have a evolving mechanism as far as Cas9 locus is concerned, or CRISPR locus is concerned. So they use different Cas and different kind of small RNA like cRNA or a combination of the tracer RNA. They do so. So this is very important. And then we have the gene specific, the CRISPR, how they do so, because this is the sgRNA, single guide RNA. This is the guide RNA which is recognized the Cas9 protein and from the complex. This complex then target the DNA, and you already heard about the PAM sequence, and have a cleavage of just upstream of this PAM sequence, and then the insertion of the sequence occurs. So this is a target specific guide RNA assembly. The case protein complex, the prerequisite to have this kind of reaction, both in vitro or in vivo. The RNA protein complex associated with the sequence specific target DNA, and the nucleus domain of the Cas protein creates a double strand break on the DNA, and during this repair of the double strand break, gene can be edited as desired. So there are a lot of different Cas9, you already heard about it. So one of the Cas12a is there, which is also a nucleus. Is only thing, the difference they have the different domains, but also they use the different kind of target sequence, either guide RNA or cRNA. Based on that, they target the different sequence, and you have other kind of multi-protein complex that may form, and this cascade Cas3 is another example of it. Although Cas9 is the most popular, widely used, and other Cas family protein also perform the endonuclease activity, and that give the diversity in the system that bacteria has against their pathogens. So guide RNA expressing construct and Cas9 expressing construct are designed as per the plan. So this is the way they usually modified. So the target sequence in the tracer RNA scaffold are hybridized to each other such that they can form a single guide RNA, and then the Cas9 is also generated. So you have the specificity coming from this cRNA locus, and the tracer RNA moieties actually give you the guide RNA aspects to bind the Cas9 more specifically, and this brings the sequence to the right place, and the right kind of cleavage site has been recognized with the help of PAM sequence, and you can have this kind of protospacer there, and in that way you can cut the desired DNA. So both construct are introduced into the cell or animal model, and eventually the cell lines or animal with edited gene is generated. So that's the way it has been modified to use as a genetic tool, which has now have only two components: the nucleus, the Cas9 nucleus, or any other Cas nucleus, and the functional guide RNA. Both go together. So how they are used? So non-homologous end joining is one of the mechanisms by which the cleavage can be introduced, and some kind of deletion may occur, some kind of addition may occur, and the mutation may introduce in this way. But if you have a targeted large deletion between two cut sites, it can be done. If you have a DNA of target and you have targeting two locus of the DNA with this Cas9 specified with the specific guide RNA, and you can introduce cleavage in the both sides, and then you have a long deletion of the fragment. So with this way, you can introduce a deletion of a particular gene locus, or you can introduce a DNA into the locus, because you can, as a foreign element, you will express another target sequence DNA, which also get cut in two places, and that is introduced as a donor sequence into the target sequence that are clipped by again another guide RNA CRISPR complex, Cas9 complex, and then it's getting inserted. So homology independent targeting integration may occur. But if you use the HDR technology to mediate the repair, you may find that the donor template is provided, and they have a homologous recombination after the cut, and you have the targeted gene insertion. It can be introduced by a single stranded ODN donor, and with the repairing mechanism in place, you can have another targeted alteration of specific sequence occur, because the donor oligo actually have this specific sequence or alteration in there. Or exceptionally, there is a single editing can be possible. So if you add a FOBC moiety fused with the Cas9, or introduce a separate component interacting with the Cas9, then the deaminase function will occur, and the C to U conversion will, cytosine should be deaminated, and the nicking will be done, and cellular mismatch repair will ensure there is a mutation introduced into that. So there is a targeted nucleotide alteration is possible with this ApoBC1 coupled Cas9 complex. So depending on the repair method, donor DNA can be sometimes contain homologous sequence, sometimes used for the editing of create deletions, knock-in, mutation, etc. The addition of donor DNA without the system. Excuse me for a minute. So CRISPR is useful for generate libraries, and from when you design a library, you can design different guide RNA against different locus for a particular gene or chromosomes, and then you have synthesized a pooled oligonucleotide with all the possible guide RNA targeting different genes in that locus, and you can produce a lentivirus with those guide RNA. And on the other hand, as I already mentioned, with this ApoB1, you can introduce these mutations in the process. This Cas9 tool, this is a genome editing tool. But there are other tools called epigenome or gene regulation tools. For example, these P300 or TATE or KRAP, these are the moieties that have a role to play in transcriptional regulation or epigenetic modification. They can be fused with the Cas9 in such a way you can score the expression of the gene by tagging some defective Cas9, which is cannot cleave the sequence but can bind only, and by that way it can bring the this protein of your interest in that locus to see the effect of this protein binding on that locus on the transcription or gene expression process. And in that way you can use a reporter system. So this is a gene of interest, and you have a fluorescent reporter along with it, and you can see how this gene expression pattern changes by following the fluorescent protein expressions by FACS-based analysis. On the other hand, if you do the screening of this siRNA against this specific locus, you can mutate or delete that specific locus, and with that you can see how the deletions of the specific locus or mutation in the specific locus of a gene can have a positive selection against a specific drug, or have a negative selection for the synthetic lethality. In all the cases, you can do the guide RNA abundance of the unselected populations, or you can recover the guide RNA library from the selected clones, and from that you can identify the enriched or depleted guide RNA and the targeted loci. So the gene function of specific loci or specific genes can be identified with this technology, like it has been done for the RNA-based technology. So this is the way it is possible to identify a gene of your interest and its function using this CRISPR-Cas9 based tool. So library construct and targeting gene of interest are created and packaged into lentiviral vector, then can be screened for efficient model generation and for the experiments. I already talking about a lot of how it can be useful. So the one of the major issue that comes with the CRISPR-Cas9 is the off-target effect. So off-target genome editing refer to the non-specific and unintended genetic modification that can rise through the use of Cas9. As you can see, there is only a small stretched of nucleotide that are complementary to a specific DNA sequence for introduction of the cut. You can imagine this kind of complementary may occur within the genome in many locus, in a non-specific way. And if it happens in a non-specific way, it can be dangerous, because if it's an off-target effect, the off-target mutations may introduce to the animal. So maybe this activates some of the proto-oncogene, or it inactivates some of the tumor resistance genes. In that way, some mutations may be introduced that can itself promote the tumor formation or cancerous growth in animals. So that is a danger that may affect. That's also there when you're talking about the gene therapy or human gene modifications. So this again can happen in the plants also, if you like to do some kind of mutations in plants and have abnormal gene functions. So abnormal phenotypic expression may happen or affect also the plants and its growth. So we all good thing have a bad thing also associated with it. So when you're talking about the specificity of the Cas9, which is sometimes not that specific. So with modification of the Cas9 and the other Cas protein, people are trying to get rid of this problem, and has been addressed, and specificity are increasing with other forms of Cas. So CRISPR.
Shubhendra,
Yes.
Shubhendra, would you mind? There is somebody to ask a question. So would it be okay if we can take the question right now?
Sure.
Okay, okay. So Sonia, please unmute yourself and ask the question.
Sonia Saraswat, please unmute your mic, ma'am. Sonia Saraswat, ma'am, please unmute your mic.
Okay, I think there is a technical problem. So, Shubhendra, you can continue. We will wait till she can get in once again.
Okay, no problem.
Okay, thank you. Thank you.
Okay, so there are different approaches I already talked about. So gene editing is only one of them, where you have the guide RNA sequence, and Cas9 is leading to a specific cleavage of the target sequence with the help of this guide RNA and PAM-directed orientation of the genes, genomic sequence, and then the cleavage occurs. But there are other ways. The gene modification can be introduced by this CRISPR-Cas9 technology. So with the development of dCas9, which is a defective Cas9, which don't have a mutation in such a way that it cannot cleave the target sequence, but it can bind to the target sequence of the DNA. So in that way, it can be a useful tool to tag some of the effector proteins that you want to see the effect on the gene locus by fusing it with the Cas9 itself. For example, you can have a trans effector protein that is by binding to the locus, activate the transcription process. You can have a epigenetic modifiers, the eraser and writers, as they popularly known as, to modify the histone modifications like acetylation and deacetylations. And by that way, in that way, they can modulate the chromosomal structures, because you know the compactness of the chromosome depends very much on the histone acetylation deacetylation status, and in that way this modifier can also alter the locus structure, the chromosomal structure, from the U to heterochromatin to euchromatin and so on. And in that way they can make a locus which is otherwise transcriptionally inactive to an active locus, or vice versa. And in that way you can see the effect of this kind of modifier on specific locus and specific gene expression pattern. In that way, if some genes are silent in one condition, you can make them activated by the help of this epigenetic modifier coupled dCas9 and guide RNA. And the transactivator domain is also there to put the transcription on. So this gene regulation and epigenomic editing with the help of this dCas9 and the modifier coupled dCas9 rather, they are very useful. And you can see that they have other applications also. For example, some of the guide RNA may be coupled with another sequence called MS2, which is itself derived from another bacterial fudge, but it's useful to bind certain protein that can recognize these MS2 motifs. And when they bind to these MS2 motifs of the RNA, they can bind in tandem, because they can have a lot of good number of MS2 motif can be cloned downstream of the guide RNA sequence. And when they fused with some fluorescent protein, you can tag the whole complex with the fluorescent protein, and you can find their locus by following the fluorescent of the complex, because the complex bind the fluorescent protein and they can find the protein complex in specific locus of the gene. So the chromatin imaging is also a very useful thing to look at. So not only that, you change the chromatin topology. For example, we are all talking about this kind of technology for here, this imaging, here this kind of genetic modification, here this kind of transactivator, the transcription activator is there. So you can use the base editing, as I already mentioned. This FOBC coupled with the Cas9 can be useful for the base editing in the DNA. But there is Cas13, which is already talked earlier, I think, which is useful to target an RNA sequence instead of a DNA sequence. And if they are targeted to a specific RNA sequence, you can also bind the RNA and make it inactivated. And also, on the other hand, this can be very useful to follow the RNA as well, do the imaging of the RNA as well, which is otherwise sometimes very difficult to do. On the other hand, the chromatin topology can be changed, because if you put two dCas9 in two different locus and some coupling protein there as a dimer, you can just fold the chromosome itself, and in that way the chromosome topology will be changed. So you can see how many different ways things can be used. This is another example of the Cas9 is already coupled with the fluorescent protein, and this fluorescent protein itself can be seen in the nucleus part. So these are very useful way to use this technology for the cell biological perspective, as I was going to talk about. And these are all the different things I will talk about in the application they have done. So CRISPR-Cas9 used for genome editing, CRISPR-Cas9 libraries for screening, transcriptional activation and repression, epigenetic editing with CRISPR-Cas9, therapeutic application is there, and the live imaging of DNA and mRNA is possible with the CRISPR-Cas. I'm going to talk a little bit of this activation and repression mechanism, popularly known as CRISPR-I or CRISPR-A. So Cas9 fused with well-characterized transcription regulatory domain using a pre-designed guide RNA. So you have the guide RNA which is recognizing the promoter sequence, but instead of cleaving it, it actually fused with this, let's say, GB64 transcriptional domain. And then you have this MS2 domain, which is interacting with other kind of MS2 containing proteins like NF-kappa B or P65 or HSF. And in that way, you can introduce the activation of the transcription from this promoter. So you can try to express a gene which is not otherwise possible very specifically with some chemical modification or any other transcription factor, because just overexpressing the transcription factor will not help you, because they can bind any other promoter and affect all other gene expression. So getting the specificity of a promoter specific expression, you just do it with the Cas9 coupled with the CRRNA, which is designed in such a way it encodes some MS2 element in it, and then MS2 element bind the NF-kappa beta protein or P65 protein and activate the transcription from the locus, whereas P64 is useful to recruit these factors and make the transcription happening. On the other hand, the repression can be there, like DNA MPTA is another repression that can be tagged to the MS2, and the Cas9 is coupled with the KRAB. In that way, they will introduce the repression of the gene expression from there. So this way, you like the RNAi mechanism, you mute a gene. So in this case, you mute the transcription by introducing this kind of technology like CRAB fused Cas9 or DNMT3 tagged to MS2, which can bind the CRRNA, modified CRRNA and tracer RNA, and have the transcriptional silencing from this promoter. So epigenetic editing with the CRISPR-Cas9 is also, as I mentioned, possible, as you have this Cas9 recruited to a specific locus help of this guide RNA, and the editor is actually coupled with the Cas9, the repressed locus. This editor will come and do the acetylation and deacetylation as required, and this repressed locus will become an active locus because the epigenetic editing will be occurring there by the help of this editor coupled with the Cas9 and as a DNA binder domain. So these actually bring the specificity to the DNA binding part, this guide RNA and the Cas9, and then this Cas9 and the editing motif coupled protein will help you the editing to do. So you can see that any specific DNA sequence can be targeted with the help of this Cas9 and the guide RNA, and depending on the moiety you like to couple with this Cas9, you may go ahead to do all the editing function that you want to do, both in transcriptional level and as well as the epigenetic level. So that's the beauty of the whole system. So when you can do that, you can really think about to do a lot of modification of genome expression, starting from the replacement of the gene with a faulty gene with a good gene, and also try to introduce the mutation in the genes. But on the other hand, you can do some kind of epigenetic changes as well as transcriptional upregulations that give you more flexibility with the modulation of gene expression happening both at transcriptional level and also at the genome level. So you can think about using the technology to tackle a lot of problems. For example, genetic and lung diseases, mitochondrial diseases, cancer, genetic liver diseases, viral infection, Huntington disease, muscle Dustin muscular dystrophy, or blindness even. So a lot of application is going on. I am sure that a lot of other talks are there that will cover this topic, so I'm not going to talk about, but I'm going to talk about some kind of imaging techniques that my colleagues in other labs are using. For example, the Cas9 and EGFP is coupled and delivered as a complex with the optimized sgRNA into the system. They will bind the sequence of DNA with the specificity of the guide RNA directed specificity, and sequence specifically enriched EGF signal will be there. If you can see the signals of the green, and in the living cell you can see where these genes is located within the nucleus. So that is a very useful thing. Similarly, the RNA targeting Cas9 is another important aspect. In that case, a PAMR is used, is another RNA sequence that will bind to it, and when it will bind only with the RNA, and this will guide you the RNA sequence, and that way you can see that because GFP fused, Cas9 GFP fused sgRNA, and with this PMR RNA, it will only guide you to the specific RNA sequence, and you can follow the RNA in the cell. So this is used to RNA localization tracking in living cell. RNA is known to be localized in certain kind of granules like stress granule, P bodies, and so on. So RNA targeting Cas9 enabled recognition of endogenous unmodified mRNAs. Cas9 did not influence mRNA abundance or amount, because in all other RNA imaging techniques, you like to tag the RNA with certain sequence that can bind a fluorescent protein. For example, if the RNA is bind with an MS2 element and then the GFP is fused with an MS2 protein, then the GFP can bind directly to the RNA. But in that way, you are modifying the RNA itself. But in this technique, you don't have to modify the RNA. So that is the advantage of the imaging of this CRISPR-based RNA detection over the other way of RNA detection, like in live cell imaging. And it can be done both for the fixed and live cells. And subcellular distribution of Cas9 was highly correlated with the RNA fish, because you can use an oligo hybridized with a fluorescent tag and hybridize the RNA with these oligos to see the localization of the RNA in the fixed cells. But that techniques and these techniques with this Cas9 mRNA visualization techniques that I've described here are very correlative, and they can be used to really point out where the RNA is. So that is a very useful thing people are using. So the other way you can really directly do the CRISPR live fish. So in the fish technology or fluorescent in situ hybridization as such, you have to have a oligos that can bind to your RNA or DNA. And based on the hybridization you have done, like the northern blot or southern blot, you only have a fluorescent tag with the oligo, and you can visualize your desired sequence of DNA and RNA inside the cells after the fixation. It cannot be done in the live cell context. But in the fluorescent guide RNA and Cas9 introduced in the live cells with the fRNA delivery system, this is already coupled complex, and you can see the DNA where it's localized or the RNA where it's localized by the CRISPR-Cas9 and the fluorescent guide RNA based techniques. So no target or off-target signal degradation is there. Only the on-target rapid stable signal visualization is there, and that's the advantage of the whole thing. So this is a chemically synthesized fluorescent guide RNA in complex with a Cas9 protein that can facilitate rapid, robust, and scalable genome DNA tracking and RNA imaging in the live cell, including the primary cells. That is really useful. That are otherwise impossible to do in the other techniques used. So one of the example is here. So this is the chromosome. You can see the telomeres are there. There is a pericentriome major satellite repeats is there, and the centromere is there. The minor satellite repeats are there. So like to imagine where these sequences are localized in the cells. So what you do, you use a Cas9, which is already fluorescently tagged with a red fluorescent, and use a guide RNA which is green fluorescently tagged. And depending on that sequence in red, you can target either of these sequences, SG match SAT or SG telomere or SG minimum satellite region. All you can target, and the photoplays are used to target them. And if you do that with this SG match SAT, and which is coupled with a green fluorescent tag DY547 and CY5 DCas9, you can see the visualization and how the signals are there. Both the signals are localized to each other, so that's why you have the marked signals very much representing both the green and red signal. But if you use some mutations or a non-specific sequence, or either the SGMAT without this guide sequence, you cannot see any signal. Only the DCas9 is there. You can have a hazy red signal, but no specific points you can localize without this one of the component. So that is an advantage of doing this kind of Cas9. This is popularly known as using the CY5 level DCas9 protein and DY45 level full length SGMAT set detected pericentriomeres, but the truncated form of unlabeled sgRNA or absence of the sgRNA failed to detect the pericentriomeres. So that's the way the techniques has been used and reported previously. Similarly, there can be a super resolution imaging can be done. You know, in the super resolution imaging, we are going to the sub-nano nanometer levels to have some kind of proximity between the signals of green and red. So you can really sharpen your localization pattern again with the DCas9 EGFP and sgRNA Alexa 4645, and they are overlapping to each other. And you have the fluorochrome added to this with the specific motif downstream of sequence of the sgRNA, and you can just the imaging and you can do some kind of analysis of the image. You can see how closely they localize to each other, how the spacing is in between. So sgRNA appended with the afterwards motif, in this case either MS2 or PB57, allow the assembly of multiple fluorescent tag on a single sgRNA molecule. So to have a decent signal, you have to have a single molecular detection of a particular sgRNA. You have to have the increase the signal, and that is achieved by the help of this MS2 motif cloned downstream of this sgRNA sequence that give a MS2 motif there, which is tagged with the MS2 GFP protein or MS2 DS red protein, and they are also forming complex with this a single sgRNA. And in the single cell imaging techniques, you can really detect the single locus of the telomere and this proximity with the mass locus. For example, two kinds of probe you are using, and you can see how closely they are spaced to each other. So this is another way helping the two locus you are targeting with two different fluorochrome, and you can see how they are closely placed to each other with the help of this. So again, in the live cell, you can do this kind of dynamic movement monitoring with, for example, MUC4 gene through mitosis. You can see with time how the locus get changed from one part to the other. And this CRISPR-based imaging enable us to monitor the position of specific genomic loci in the nucleus during the cell cycle, and simultaneously label multiple positions within the same green locus that might help to characterize the local component compaction state at the labeled chromatin segment. So these are all very useful techniques. You can see that how the prophase, metaphase, anaphase, and telophase phase, the nature of the signal and the number of signal actually also change. So these are all the applications that are there with this CRISPR-Cas9 and other Cas system. There is a very interesting as development also there, which called light activated CRISPR or fast CRISPR, which is nothing but a target RNA region which is coupled with a protector oligonucleotide. Till the protector oligonucleotide bind to the guide RNA, it will not be active. But when you expose the cells, let's say with UV light, then this fragment will get cleaved. This protector oligo will get cleaved because it's a photo cleavable moiety is there. And once they will cleave, they will remove from the guide RNA, and that guide RNA will become an active guide RNA now. So this is a CRISPR off state, and this is a CRISPR on state. And so by just exposing the light to your system or hitting with a specific light signal in the system, you can specifically keep the gene on or get the gene on at a specific time point or specific requirement, and that way you can activate the CRISPR RNA as well as the targeting of the specific sequence with this RNA, and you can either desire the cleavage or you can desire the modification as per your requirement, and in that way the CRISPR locus targeting can be achieved specifically with the help of a light. So this is a very interesting system because you like to take your gene on and off at a specific time point, and that will help you to get it. So with that, I would like to end, and I try to cover all the possible methods that are now developing with this technique. So I thank again my lab members, Saurabh, Samantak, and Ishani, who did a good job to gather all the informations that are required by me to make these presentations. And I hope you have enjoyed, and I'll be happy to try all the questions. May not be have all the answers for your questions, but if you have any specific question, I'll try to answer. And if possible, I will probably also try to answer some of them by emails or other thing way if I not able to do so right now. So let's start with the questions.
Yes,
so
thank you. Yeah, thank you, Shubhendra. That was a real good session, and it was slightly different from what we heard earlier. So yes, we do have some participants raising their hands. So I think Sonia, okay, I can.
Okay, very nice.
Okay, Anu. Okay.
Yes. Okay.
Yes, Dr.
Okay,
Anu, please go ahead.
Sir, I have just a basic 2-3 questions. One was you talked about this light activated protector system. So
Yes.
that means it cannot be I mean, it could be a on-off system, or once the light will fall, it will cleave the protector and will no longer be available. So once it is on, it will remain on.
Till now, it's once it's on, it will be remain on. So maybe people are trying to get it the on-off system as you are talking about, but at the moment, the techniques allow you to put it on as your desired time or in desired place only.
Okay.
But it is not going to get off after that.
Yes. Okay. And so next question was like when we are targeting the RNA instead of DNA, what about those PAM sequences? Will they be present on the same sequence where the guide RNA is binding? Because otherwise it is opposite. Guide RNA binds to the complementary sequence of where NGS is. What about the RNA, the same sequence?
Yeah, that's why they are sometimes called these PAM marks. So they are actually provided as a separate sequence in certain techniques. So that oligo actually supply the PAM sequence that of desired sequence that will bind to, and but only the complementary part will recognize the RNA part. So that way they are avoiding this kind of misrepresentation or misrecognition. But there are other techniques like this Cas9 that you are already heard about.
Yes.
They are using a different way of doing it. So they are providing the same RNA, single RNA to get it, but the Cas9 is Cas9 is different from Cas9, so they are doing this way.
Okay, sir. So there are so many systems. So how to decide which one works better for your host?
Yeah, so it depends on the application you would like to do out. So if you try to follow the RNA, your choice would be either Cas13A or using the dCas9 with this PAM mark that are earlier developed.
Okay.
So that will give you an RNA to target with that. But if you like to do a, let's say, genome tagging, but not cleavage, like activation function of transcription or deactivation function of transcription with the eraser or writer with it, the chromatin modifier, I would say. So this can be achieved with the help of the dCas9, because this is a defective Cas9 which cannot cleave the sequence of DNA. But it can used to tag the specific sequence with the help of the sgRNA that you are going to introduce also. Okay. So with that modified dCas9 coupled with different transcription activator modifier or chromatin modifier, you can really achieve the desired activation of the genes of your choice.
Okay, sir. Thank you so much, sir. Thank you.
Okay. Thank you, Dr. Anu. Sonia, can you unmute yourself now? See if you can, or you can type your question. If you cannot, you may type your question in the Q&A box, and Dr. Bhattacharya will be able to see it. So, unless if you have any other problem, Sonia, are you there? So you can, I think you have a technical problem in unmuting yourself. So why don't you type the question, and it will be easier for us to access it. Okay. So I think Sonia has a problem because she could not ask the question earlier even. So meanwhile, by the time if others have any question, I can just put forward some question, one or two questions, Shubhendra. It is just a technical question once again. I'm just trying to understand when the tagging is being conducted. See, we are trying to understand a technology wherein the genome is going to be either modified or it can be cut at our own desired position, because it is required, because we need to cut it. Now, that is one aspect where we did not think about any tagging, or it was simply cut, and downstream we checked whether the cleavage has occurred or not. There will be some way to test that. Now, when you are going for a different approach. Now you are trying to tag something which is visible. So with a reporter, we are visible. This visibility is required also at the level of cell biology. It is very important because you need to trace it. Now, when we are going for an approach like with Cas9 and a kind of a fluorescent protein or whatever, means I think it should only go with Cas9 if I'm correct. Should it go with the normal Cas? Because then it will
because normal Cas is there, the cleavage will
the cleavage will. All right. So if we are going for Cas9 and tagging it, other than localizing the RNA, what extra information can we extract? This is first question. Whether this after hybridizing or after joining to the desired position, then only will it fluoresce, or it is going to be having the fluorescence from the point it is entering. Means I am wanting to know what extra information we can gather other than localizing it, and whether the fluorescence is going to be limited after hybridizing and not before that. Then the entire system will be glowing. So there should be some way that only upon joining it will fluoresce, and then we can go ahead and do the next cell biological work. So did you get my question, like whether there can be any noise?
Yeah, there would be noise. There would be noise, and that's the difficulty still with the techniques. So because you have all the let's say fluorescent coupled Cas9 is already there, and they will be moving around, and you can see that. But the thing is that unless they are basically bind to a specific locus and get concentrated there, the whole signal will be very much, I would say, mild.
Diffused. Or maybe diffused.
Signals. And only when they bind a specific sequence and they get enhanced, then only they will be activated. But there are way also people do it, because there are some kind of coupling method when they are only form a complex, they only form the fluorescent part. Like they are also achievable like that. The two part of the protein come from two different components such that they only join together, they only diffuse. So that is another way approach people are trying to adopt. But the most interesting is that using the co-localization of two kinds of signal. One, let's say green is coupled with the Cas9, whereas the red signal is coupled with the RNA itself, it's sgRNA. And then you have the both signals together, means that you have the right signal.
Yellow,
So that's the way people are trying to get the specificity of the whole system, whether we are seeing it. But from the cell biological point of view, it is important. But when you're talking about the gene regulation and screening based on that, you just try to target different genes with this, let's say, gene activator function. And you see by activating a specific genes of your interest in specific context, whether you can able to get the phenotype of that particular let's say growth or drug resistance or anything. So like the RNA, I think you can only inactivate the gene, but in this case you can activate as well. So both the activator and inhibitor signals can be achieved with the help of this CRISPR-Cas9 based gene. The disadvantage is still there with the non-specificity is there still. It's a question, but with the modified Cas9, I think they will able to address the issue of the non-specificity.
I think so yes, then that explains. And when it was going to bind, then the signal will be more specific. Although we can think that there would be a noise, but that specificity will itself speak that what is happening at the cellular level. Yes,
Yeah.
that explains. Yes.
I think
Yeah.
Rudronin has a question. I tried
Yes.
to answer.
Can you
He is asking,
please.
is it possible to use a bulky complex like Cas9 guide RNA to enter inside any organelles? To image RNA inside the organelles like mitochondria, ribosome? The answer is no, because mitochondria don't have the pore size that to allow the entry of the Cas9 guide RNA complex inside the mitochondria to see the mitochondrial DNA or mitochondrial RNA inside the mitochondria. But as far as ribosome is concerned, you can do it because ribosome is present in the cytoplasm in a free way. So if the ribosomal RNA part is exposed to certain extent, then you can do it. But unfortunately, most of the ribosomal RNA is wrapped with proteins. So you hardly have any ribosomal RNA exposed on the surface that can be bound by the Cas9 guide RNA complex. So whatever you can target the ribosomal RNA with this Cas9 guide RNA is during the formation of the ribosome phase, which not happen in the happen in the cytoplasm, in the nucleus itself rather. So it is possible to technically possible to do the ribosome imaging, but not for the mitochondrial.
So is this technique, especially for RNA, is it possible for all type of RNAs, or there will be only some specific RNAs that can be better substrate?
Yeah, I think this is a very interesting question. I don't think that can be answered any RNA in that sense. For example, tRNA, for example, they're heavily structured. So any RNA which is heavily structured because of the help of the other element present, and that's the ensure the hybridization of the RNA within the RNA sequence to fold and create a primary and secondary and tertiary structure of the same. In case of ribosomal RNA, tRNA. So in that way, some of the mRNA UTRs or particularly the 3' UTRs are very much structured. They have a lot of structural element present that fold them back on each other. So maybe targeting those RNA with the Cas9 complex is not an easy thing because you are only relying on a small stretch of RNA present on the guide RNA to bind it, and the Cas9 complex is there. But yes, the efficiency of guide RNA mediated Cas9 tagging to specific RNA locus depends on the RNA, very much on the RNA itself and nature of the RNA itself. Not all RNA will be efficiently targeted by these techniques.
What is your view on microRNA in that case? Any?
The microRNA is very small, so I don't think that you can really do targeting of the microRNA, mature microRNA itself by the guide RNA Cas9. The only way you can do the targeting with the precursor microRNA
upstream, so right?
Yeah, so they are bigger in length, but the problem again, they have a structural element present in them that let's say stem loop like structure and so on.
Yes.
So I don't think this Cas9 guide RNA would be good enough to target those complex structured RNA because they don't find the specific sequence required for their binding because they're already part of a structure that is stable enough to be melted by the Cas9 and Cas guide RNA complex. So in that way, they will not be able to do so unless they are targeting the genomic locus where the transcription starts. So that is possible.
Yeah. Do we have any other question? Anybody raising hand can raise your hand or can post your question.
Rudoni's question has been answered.
Already answered, and I'm not sure whether Sonia was trying to get in, but did we get her finally? Maybe not, and she has not posted a question as yet. So okay.
And we have RK Gupta.
Yeah. RK Gupta. Okay, just go ahead, please. Unmute yourself and ask the question. Hello?
RK Gupta, sir, please switch on your mic and you can ask your question. And you have to unmute your mic to ask the question.
Please. Yes, please unmute. You can go to the left and check the microphone symbol. Maybe they're not getting it. Is it
Are
possible
you
for
good,
you?
sir?
Yeah. Is it
You
possible
can
for
just
you?
unmute your mic. Otherwise, you can put your question in the chat box.
Yes, we have time, so you can take we can take the question if you type it. We still have few more minutes, so no problem. There is a problem in locating the mic, I guess. It is in the left side. So let me see if there is any other way they can ask. Okay. Because there's having some problem maybe. So I think, Shubhendra, since we have a few more minutes remaining, I just want you to, it was a good session, absolutely something new we learned. And of course, it was a bit at the cellular level. So mostly it dealt with the scientific aspect more. And yes, we can translate that and try to understand whether we can bring it out at the application part. So that is definitely, we will always go for such research, such technique, evolution that can be finally be applied to our society for the societal benefit. Now, what is your advice to the young and all the faculties that are there on the other side of the screen, because you have also gone through a long journey from working in the institution and then embarking into your research area dealing with RNAs, and now and you have gained such popularity on not only on your scientific endeavor but also on this this prestigious and commendable achievement through the different awards. What is your view for the faculties and the students? How should they prepare themselves for attaining these heights? Because that would be a very interesting and very helpful suggestion or advice from your point, from your end. That would really be very
Thank you, Jyoti. I'm not sure I answer it because it's very much specific to the individual. So that's
that is definitely yes, that is
I'd
true.
like to highlight few things. For example, the question that you are trying to address during your PhD and postdoc is very important because you have to think about, let's say if you are doing a PhD at the moment and you are planning to go for your postdoc, you have to think few things you have to keep in mind. For example, when you are talking about the postdoctoral options available, so try to find a postdoctoral questions or laboratory which is addressing some kind of, I would say, futuristic question. Sometimes you go to a lab where the field is a little bit more saturated already and you hardly have much space to grow. Whereas there are few new challenging things coming up which are not addressed properly. For example, this CRISPR field, of course, on them. And there are a lot of possibilities there with your research questions. So nowadays, I would say the thing is that the more and more involvement of mathematics and I would say quantitative aspects of biology is emerging. So you have to think about when doing a biological research, think about the evolution of these quantitative biology aspects in next 5 to 10 years, which will going to govern. So when you getting back after your postdoc, you have to have a relevant question with you to get your grant and everything to be awarded in proper time, because without proper grant you cannot do much progress. And to get a proper grant, you have to have a much interesting questions that not only relevant from the scientific point of view or contemporary point of view, but you also have some relevance in the disease biology or some kind of application orientation as well. Because without that, I didn't think you have much choice worldwide, not in India, even worldwide, to survive, because you have to have some relevance of your research to the society in that sense. So keep this thing in mind when you are deciding your postdoc lab. That's my first suggestion for the PhD student and the early postdocs. If they're going abroad and getting abroad, or even you can do it your postdoc in India itself. That's nothing much less in that way. But you have to ensure that you have a good question in your hand. So don't try to just to get ahead with your PhD experience a little bit strengthened in that way. Try to be a new field. Try to be in a field that will help you to nurture your own lab in future. So that is the first thing. For the young faculties, I think I would suggest that they try. The first thing you have to ensure that you have to ensure some grant. Of course, you are all doing that, but in the sense you are also trying to I would say diversify yourself a little bit. Maybe you are getting a grant for certain one thing. You have the obligation of delivering the thing on time on the specific addressing specific objective of it. But try to be a little bit diversified within that grant also. You don't have to have the I would say you have the luxury to get really different from what you have written in the grant, but you still have the flexibility to get a little bit different you have done within the grant to address few new aspects that were in your mind. You cannot propose it because you have no tested results or evidence to support that hypothesis that you had. But maybe this is the time when you have this grant in your place to try to address a few of these interesting aspects. So when you do that, then you get some new leads that may not aware at that time how it would be leading you to, but that would be very useful. That actually happened with me, and that's why I'm suggesting it, because whenever I get a grant, I honestly speaking hardly address 50% of the what thing I write in the grant. I always try to use the things to get a new things to be done, and where you can have a lot of interesting questions. So that's led me to go into this extracellular vesicles biology to certain extent, which was never in my mind and never in my plan. But gradually, I find that is a very exciting thing to look at. And the question related to that, how the extracellular vesicles are used to carry small RNA between the cells and make the gene expression alteration there. So that are there. The other aspect for the young faculty is to build your team. So because the building of your team with 1-2% and then gradually grow on that is very important, and you have to find the right person at the right time, because if you don't have good student and postdoc with you at the early days, then it will be a problem. So be very specific with your recruitment process at the initial phase of your journey, because that will decide how you will do in next 10 years. So try to get people who are smart enough, they are hardworking, as well as they have the commitment and passion. Both are very important. So if you can build your team with those people around you, you will be successful.
Yeah. So, this was really important, because all of the faculties and the students, they are actually trying to prepare themselves for the next phase of their life. And your suggestion, like I also feel that you have rightly pointed out that do not go for a research work or PhD just for the sake of the degree. There should be some very pertinent question, and that question should be such that it is slightly making a difference. It should mark a difference, and that difference will actually be judged in such a way that it has application at an application point of view. Because only basic science is definitely required. That there is no, it doesn't. It is absolutely goes without saying that without basic science we cannot move ahead. But I will also, taking your advice and to the faculties, I would also ask the faculties to encourage their students. That is more important, because the talent pool is also lying in the student community. Yeah. So if all these type of workshops, programs can drive that question into the faculty's mind, and also there are some students, I think this message will be good enough to take them forward, at least a step forward, to make them infuse within the students' community, their mind, some good questions. And for that, they need some reading, some awareness, and attending these type of programs that can make them at least curious to find out some unknown aspects of the scientific, all these scientific information, and try to. It's not possible for you to answer because you would be requiring experienced person to guide you. You need collaboration. You need discussions, but eventually you will benefit. And so try to get some good questions, and those good questions will really be the first step to move ahead and get all the accolades.
Yeah, only the proper questions will lead you to the proper answer. Only that's the point.
Answer, right. And I think that is one very piece of advice that Shubhendru has made today. Prepare yourself. It is okay that you might not be able to connected. So it might not be directly related to the topic which we were discussing, but at least these programs will also let you prepare yourself as a good teacher, and a good researcher. So I think we have covered quite a bit of our doubts, and we had a really good session. Dr. Bhattacharya, thank you very much on behalf of our university, and we will keep in touch with you, and our participants will also get in touch with you. And yes, definitely. After connected or Shubhendru got disconnected, I'm not able to understand. Okay.
I'm hearing.
Yeah. So my internet is pretty unstable, so I'm not able to keep in line. So anyway, we will all be in touch, and I also I extend my invitation to Dr. Bhattacharya to come over and visit our campus. And thank you so much, and it was a really great session. And with this, we end our morning session, and we will